Нажать, наверное, понятно, да? Все, наверное, тебе нормально, да? Все идет, да?
Коллеги, скажите, нас слышно в интернете?
Да, слышно, все нормально.
Хорошо, тогда начинаем, но у меня будет на английском лекция, потому что это iBrain, но если что, можете что-то переспрашивать, наверное, или в вопросах. Okay, so our lecture today is gene, cell, brain, and behavior. And while driving here, I noticed that the real name of this talk is announced by in our chats was gene-brain behavior approach in humans. And that's a little bit surprised me, but what I will be talking about is the translational approach, how we can probably translate the data obtained in animals that what you already learned through the courses to humans. And I will also use some examples in animals as well, just so be with me that this lecture will be not only about humans, it will be about animals as well, and the approach in general and how it can be applied to humans. And the framework is very, very evident and already in scientific field for many years, many 10 years, I think. And that we want to understand the human being or the other organism on multidimensional approach. We want to kind of combine in the united picture what's going on at the genetic level, what's going on in the cellular level, and the cells connect in a very unique organ is our brain, how the systematic features, functions takes within our brain, and then how it's translated to human behavior, to animal behavior, and to psychological functions. As you can see, it's from one side, we understand that the whole organism contains all these levels, but how they interact and each gap between the each kind of level is not very evident. It is well-known physiological problems from the times of Descartes that how our physical, biological process relates to psychological functions, but that's even in the level of the organism, within the biology, each level of as we increase complexity, we don't understand the mechanistics, the fully understand all the mechanistics stuff going on there. But we are getting closer to this. I will just point you to some gaps and to some progresses within this framework. So the easy, and certainly all this level, they interact, they on the different ways, and that's creates another complexity problem. So what I will be talking to you is the basic concept about from the school time, I think, about what's we can see on genetic level, biological, and other things. Then about interspecies translations, some approaches to humans as candidate genes and polygenics course. Then I will talk about genetic disorders, autism, and then just the future perspectives. Here you can see that what is our genome contains of is DNA, and DNA has a helix structure, which due to chemical compliance can be connected that nucleotides such as guanine binds to cytosine, adenine to thymine, and then we have a whole set of the DNA which contains different letters, such as nucleotides within the whole DNA sequence. And also very evident that we have 24 autosomic chromosomes and one specific to males or females, X and Y chromosomes. And so that was kind of a reminder and probably to interact with the audience, but as far as most of you are in a Zoom, I think it might be harder, but just to remind you how we call the chromosomes and how to read the things. For example, if you find some disorders or something associated as like 15q11, that's mean that it is 15 chromosomes. 15, it's, as you can understand that in our cell, the chromosome is not aligned in particular orders. It was kind of arbitrary named as a name, but it's not fully arbitrary. It just relates to the length of the chromosome. So this is 15 chromosome. Q means the long arm because the chromosome has such some kind of bands in the middle, about in the middle, and that is on the band and in which particular bands. But certainly within this band there are many nucleotides, so that's not very precise naming. And also very often we have the same name for gene and the protein that it is synthesized. How we can differentiate it? Usually that we have we use italic script when we refer to gene, and kind of normal script when we refer to the protein which produced by this gene. Also to differentiate between, we have some genes that is conservative among the species that is the same in animals and in humans. And usually when you talk about humans, you have bigger letters, all the letters capitalized, and for animals we have small letters. But not everyone now follow this kind of rules, but just for easy for you to kind of orient in manuscripts when you read it. What we have now, what can be changed? Because from one side we have similar DNA among people to the huge extent, more than 99%, but and even with the different species. But from the other side, we are different, and the difference can occur in different ways, but more common is when you have just some region in the DNA replicated and repeated. It's called CNVs, copy number variation. You can have some part of the DNA pattern just replicated more time that it was in an original thing. And other common kind of mutations, if it's functional or just polymorphism, is CNPs is a single nucleotide polymorphism, is if you have just particular nucleotide changed within the DNA. So this point mutation can be have different meanings, functional meanings. So if we just change one letter, we can have so-called missense mutation, which changing one letter usually just change one amino acid within the protein because of the changing in the kind of recipe for this protein. But if you have, you may have a small frame shift insertion. So if you have a small insertion here, that it's or deletion. If one letter is just deleted, one nucleotide or added, And that is cause more dramatic changes because all the other triplets which codes for amino acid will be changed, and the protein structure starting from this point of this mutation is changed. And you also know that correspondence between the DNA sequence and amino acid is a triplet, and some triplet, most the triplets codes for amino acid, but some triplets called the stop codon. So it's code for that the protein ends. There's a translation have to be ended. And in this case, when you sometimes when you change single nucleotides, the triplets which codes for amino acid became a stop codon, and in this case the protein stops kind of creating from this point, and it's also maybe more dramatic changes. So SNP mutation can have different functional meaning depending on particular places, on particular changes, and so forth. And that's like just representation of it, but the letters is a little bit changed. So you can see yes, here that the DNA, which is transcribed to RNA and then to protein, and triplet codes for the particular amino acid. And when you have missense mutation within only one letter, the C changed to D, we then have completely completely different protein, not fully created, translated. Yeah, and that's a nonsense mutation. So then you have stop codon frame shifted. That's all the frames that shift, and that is but the different amino acid starts to be translated. Okay, so that we talked about the most known part and how the gene translates to the protein, and here we know quite a lot about the mechanisms. You can, I think we will not stop here, but you can look at the YouTube, and there is a very great videos how the DNA is at the beginning transcribed to RNA, and there is also some particular parts where we can influence that it's alternative splicing and so forth, and then created to the protein. So what is transcriptome then? So why we need, like, why we don't just start to create proteins from the DNA? The DNA is usually holds in a kind of passive form, which is we can't read the recipe that is written there. And in order to start to create the protein, we need to kind of unfold this DNA and make the RNA, the transcript that says the proteins, the ribonucleic acid, which kind of holds the recipe for the particular protein. So in this case, the transcriptome is the set of the this RNA, which represents which particular genes are expressed within these cells. And it is important because, you know, we not all the genes are expressed everywhere, and in the particular cells, the particular protein is created, and that's important to check and to understand. We can also look at the anatomically comprehensive atlas of the human adult's transcriptome, and that's in which particular parts of the brain which genes are expressed, and they will and you can see the genes which is grouped particular kind of function, not functions, but particular pattern of expression. They presented here in different colors, and you can see that in different brain regions, the different patterns of gene expression occurs, and which we can visualize, test, and when you create some of your hypotheses, you can see how brain is different, even in the gene expression level. And now we got already to the most complex part of this diagram, is that it comes to the brain. And okay, probably some questions to the audience, but like, how many genes do we have in our genome? About how many? Yeah, so it's although our DNA is contains milliards of nucleotides, the parts that is codes for particular protein and calls the genes, that was a kind of surprising finding in human genome projects, that we have only like kind of 20 or like 15,000 genes within our brain and within our organism, the humans. Do you think so? How many genes, or how many of these genes are expressed in the brain? Like most of the genes, indeed. And but also all the genes that are expressed in the brain, they express not only in the brain, they express in the other parts of the body too. And if we will look at the genes which is elevated or uniquely expressed in the brain, it will be only quite a few genes, like only detected in the brain is about 200 genes. So that is not expressed in any other of the part of the cells. It's also kind of very interesting, but many genes are expressed in the brain, most of them, but they are also expressed in the other parts of the organism. So it is interesting to know. And also each processes on the synaptic connection and neural, they are coded by many genes. And so just to think about, so that such enormous complexity of the brain, which contain how it's created. So our genome is also very complex, but it contains only like 4 different types of nucleotides, so 4 letters. But then when we talk about amino acid, it's already 20 and millions of the protein. But then we have billions of neurons and also glial cell that is interconnected, and they are not static. They create the dynamic world of the connections, of the differences. And so just it's enormous complexity. We can't really deal it with the common understanding, and most of the conference now, they also that relates to neuroscience, they state of this big data problem, like, and how we can deal with all this information. But there is a way, certainly the methods, how we can disentangle all this complexity, try to simplify it some parts. But just understanding of how it's enormous complexity, I think makes our heartbeats quicker. And so what else we have that just makes it probably what we can focus more if we talk about humans. There is some part of the human accelerated regions, and that is uniquely expressed genes for the humans, and that's also pretty few. It's also about this orders of 200 identified so far. So much of the genes which we contains, they both have some conservative expression pattern and contains within the different organisms. So we can like, you know, depending on the how far we are in the evolution from the particular species, the less we have common genes. But we have, as I can demonstrate, to have a lot of genes in common even with insects. And this gene is called homologous. So they are conserved and the sequence of the particular DNA structures that is conserved and present both in our ancestors species in humans. And that's one of the examples is the cloak mutant Drosophila, the most commonly used species in genetics. And very, very far ago, even in 1971, Benzer showed, noticed that his Drosophilus has different patterns of activity within the day. When they're most active, it's presented here in the dark parts, and they are less active in the other parts. And if they kind of align on this, and then you have the 24-hour cycles. So most Drosophilus active in a particular part then of the day, then inactive in the other part, and it's this pattern that goes on through the days. And Benzer also used particular induced mutagenesis in this Drosophilus, and he found that there is a couple of different types of mutants in this Drosophilus. So some of them are rhythmic mutants, they express a random pattern within the day, and but there is a short period mutant that have a 19 hours of this cycle, which doesn't relate to the typical circadian rhythm of the sun and light in our world, and long period mutants, which has a longer period of the stuff. And by at first identifying this type of flies, he then identified a particular gene which was a mutation and which caused this different pattern of this circadian activity. And he called it period gene. And much later, already in our century and the end of the previous century, but the psychiatrists, American psychiatrists, noticed the particular pattern of activity in the day and night in their patient. They came mostly because of the depression symptoms, but as they identified that within this family, they tend to go to sleep much earlier, and because they can't stand longer and get up very, very early as well. And that creates a lot of social problems and health problems as well. And when they put them in a free run environment without even the changes in day and life cycle that they can't be can be seen. They identified also the particular pattern of sleep activity and also the body temperature, which because all our organisms change within the circadian rhythm cycle. And what was very surprising, but probably not very surprising, but they have a smaller cycle and this changes the shift of this phase in the circadian rhythm, and they call this familial advanced sleep phase syndrome. So depending on the time of the day, they constantly from day 1st to day 19, they shifting their cycles towards getting to sleep earlier and getting up earlier as well, without even any external cues about this. And that's how they again about the particular symptoms that they have kind of shorter circadian cycles. And not surprisingly, that they identify the particular gene which is conservative between the Drosophila and humans, and creates a particular part of this particular pattern of the morning larks, so-called shifts. And certainly it's the kind of extreme cases, and we all, I think, on the spectrum, so between different kind of inner cycle of this stuff, but the change it for 1 hour creates indeed the huge problems in life. And they identify the pattern of inheritance for this. That's just again the reminder. So if you have autosomal dominant, that's the affected gene, you will have affected child. And if you have autosomal recessive, that's half of your offsprings will halt the mutation, but the affected will be only one child. And here they also identified the pattern of inheritance for this particular syndrome, and that's how it's usually presented in the familial and histograms. But I think I will not go too much into details for this. And certainly if we have advanced sleep shift syndrome, we should have delayed sleep phase syndrome also as well. And indeed, the gene close to this previous gene was identified, its function, and depending on the polymorphism of these genes, the people have to indeed the delay phase or longer circadian cycles. And that was identified a little bit later, I think, in 2010, but I don't see the date here, sorry. So that was just to kind of sum up that there is a spectrum in humans that have a different circadian cycle, which can be shorter or longer, and that creates sometimes a problems called advanced or delay phase sleep syndrome. And these genes, again, this was common among the species, and recently it's in 2017 even got the Nobel Prize for this. And what is also have to be kind of emphasized that as you can see that it's really like, I mean, a long periods, like 24 hours, how organism can kind of contain this long oscillation, because so many things go on within this 24 hours period, but they still have to be kind of constant and contain these cycles. And what they found, it's even on the level of the gene to from DNA to RNA and protein translation and transcription. So this cycle of how the gene starts to express relates to the feedback loop. So here is an example that you have a period gene. It starts to express RNA. From RNA, you see the protein, and as the proteins gets more and more, they bind with another protein and stop the translation. They close the expression. They kind of close the DNA, and they stop to produce RNA and the protein. And when it stopped to produce, no proteins is creating here, and then this unblocking of the particular gene region occurs, and the cycle starts again, that it starts to express the gene again. So, and this cycle is that long, so within the 24 hours, it's not like seconds, not minutes, not hours, but like hours, 24 hours, and around this. It's how the gene expression controlled within this feedback loop. Okay, and that's circadian pacemakers and Drosophila, and in humans, they kind of correspond. What is more kind of surprising and interesting, that was also wanted to emphasize that even if we call to prokaryotes, cyanobacteria, which their life cycles is smaller than 24 hours, mostly. And what was found, and also about the same time as they found about the advanced sleep syndrome, that even in these prokaryotes populations, there is also circadian oscillation. kind of how it is possible. But the thing is that what we tell about the life cycle in the prokaryotes, it's not that the cell dies and the like offspring starts to live. The prokaryote cell, they just dividing, so they have all the genetic materials within them the same, and they kind of start to live, but like in a double kind of spaces and so forth. And what they found that even if they put in a light environment, so there are no change in light and dark, and check the expression of particular genes, the promoter region, they found that expression of this gene change within the hours in particular cycle. Some of them gets within the 24 hours gets smaller expression level and then occurs again, and others on the contrary. And even the speed of the kind of this population growth is different when it's in a light or in a dark environment, and it's changed even they put in the light environment, and attenuates only like after 3 days or so forth, even in a light environment. So it's not the changes to the environment, but it's some endogenous process that oscillates within the cell populations, even in prokaryotes, and it regulates how the cell divides. What is it, adaptive or not? And here you can see that the process is adaptive. If we put the two different groups of mutant cyanobacteria, they also find the particular gene there in prokaryotes, which regulates this cycle. And if we put it together, like in the beginning, it's like a fraction of this C28A is a mutant cyanobacteria with the cycle of the 30 hours, and they have the proportion of about 60% among the other wild type 25 hours, 24-25 hours circadian rhythms. And if they put it in the environment with like our normal environment, like light and day, light and dark, 12-12. So within some generation, like you will see that the fraction of this mutated cyanobacteria decrease and also fully dissipated. But if you put the same proportion of cyanobacteria in the environment with light and dark cycles 15-15, you can see that wild type cyanobacteria dissipates, and the dominant cyanobacteria is the mutant one. So it seems, not it seems, it's like evident here that the particular circadian ripe in cyanobacteria, in this microorganism, is adaptive to the environment. And it's kind of fascinating, I think. Okay, so again, so that's an interesting translational thing from the different spaces. And here we can talk about simple approach and history, how we can deal with connection between gene and particular function here. Okay, it's partly in Russian, but that's fine. So how we can connect the gene to the psychological function. It's simple. Know that you can have some DNA sample and to check how it relates to particular function, like, and you can choose some any kind of gene and forms, and as well as you can separate the people, like as they depressive or non-depressive, or you can separate the groups but having particular polymorphism or other polymorphism on the genes, and compare the population. But what is the problem here? Among that, we have a huge number of genes and many numbers of psychological traits which we can relate. The other problem that we even can't fully say that it is causal relations now, but we want to have knowledge about the mechanism, not to relate. Okay, this genes is related. We found, for example, we find the correlation, like the people who holds like long arm of particular polymorphism relates to particular function, and we found it statistically. What does it mean? From one side, you can say that it's just a correlation. You can say that it's just a correlation, but kind of intuitively you think, okay, if it's gene related to psychological function, that's the gene cause this difference in psychological function. But this is not the case, because you can have gene links between the different other genetic region. You can have some particular thought factors that cause this relationship between genes and psychological function you are interested in. So to be more close to the causal relationship, which we really want to know, we need to try to base our search and our correlation on the particular biological mechanisms. And here I will present you the study on the serotonergic genes. And you know, there is in the cell you have many agents, and for the cell to start to oscillate or to change in the postsynaptic kind of currents, we have receptors which binds to particular agent, in this case serotonin, and how many the serotonin available within the synaptic cleft is regulated by the agents which destroy, dissipate this serotonin, and there is also reuptake transporter which catch the serotonin and returns it back to the presynaptic cleft and postsynaptic. I will change, but this is postsynaptic cleft, and this have receptor, and presynaptic cleft which has a reuptake transporters which catch the serotonin and goes back to the presynaptic neuron that doesn't let it to reach the receptor. And for each of this part of the scheme, we have a particular gene that regulates the amount of receptor on the postsynaptic cleft, the amount of reuptake transporter, and the amount of particular agent which destroy the serotonin in the cleft. And here I will talk about time, and later we'll talk about the type perception. And it was shown that a particular pharmacological agent which influence the activity of serotonergic system, they change the perception of time. So when we were hypothesized that if we took the same genes which regulates the processing within serotonin gene, then we will have the changes in the within individuals in time perception. Okay, and now just an example of this one very well studied gene, and which is very known in the candidate gene research, is gene of 5-HTT LPR region and serotonin transporter gene, which responsible for reuptake of the serotonin within synaptic cleft. And these genes contains particular structure, which is all the genes that has promoter region, and the promoter region can be either long or short. And depending on this length of the transporter region, although it's transport region and seems not to be functional, but it's appeared to be functional. And if you have long allele, we have a lot of transporter gene, and if you have short allele, they have just a few transporter protein. And that certainly relates to how the serotonin system works within the brain, and that's how widespread modulation of serotonin within the brain we have. One of the seminal work about the serotonin transporter gene, it was found that it's interacts with the life events and environment. As all our genes, they certainly influence and interacts with the environment. So no particular pure genetic effects of any genes and any disease can be found. And here, when the authors from Caspi from States, he took the group of the people with short promoter region of the serotonin gene, and it's divided on the percentages of major depressive episodes. And here on the x-axis, you can see amount of the life events, stressful life events that they experience through their life. And as you can see, then they have a lot of stressful life events. The people with particular genotype, they have a much more depressive symptoms and develop the depressive disease. But for the genotype of the long promoter region, life events doesn't have much influence, and they didn't develop depression. But you can see that about just a warning that in candidate gene approach, it's very hard to get the significant findings within the limited sample sizes. And there is a lot of study that may not replicate this finding with different approaches, but there is also a bunch of study who replicate it, and there is kind of still a debate whether it's a true or not a true finding, and different groups are arguing about this. But in even meta-analysis, they contradict each other. Just to put a black dot on all the field, that there is still a lot of debates about different findings. And also it relates, this serotonin gene function relates to the activity of different brain system, and in particular the interaction in cortical limbic processing. And that was found that long allele, they have a better connections between this region than the short allele carriers. And that's also can be intermediate phenotype, which relates the finding of that's why they don't that prone to the stressful events. But also to tell something, and that relates to the total harm avoidance behavior, but let's make it out for now. And what we say that just short allele seems to be more prone to life events and more prone to develop depression from the one side, but from the other side, the other studies showed that they generally more prone to environmental influence, and even to the good one. And in the good environmental condition, there develops better cognitive abilities. So this gene seems to be related to how it's interacted with the environment, and but also was related to impulsivity and different functions. And what we can see, so from one side, it can be related to one common particular mechanism, which is good, and which I believe that we can work more on this. But it's also the other important things to know, that genes have a pleiotropic effects. So the same gene can participate in many other many different functions, and which seems to be not related for now. and each psychological function relates to many genes. That makes the situation very, very complex again. And go back to our research on time perception that was based on particular biological grounding that, as I already mentioned, this was a psychopharmacological finding about the serotonin influence of time perception, but there was also a hypothesis and finding that dopaminergic agents, pharmacological, also changes the perception of time. But as we now know, and it was confirmed also with one study with Dr. Portnova, who works here in this institute, that dopaminergic genes affects more short duration perception, like within 1 second, and serotonergic genes affects more on a longer stay, several seconds perception. And here again about this our approach, and that's illustrates a little bit that we don't took only one gene, but we took now here to a few genes, only 3, to create so-called polygenic scores. So we kind of coded from the literature the effects of the genes and polymorphisms which have on the activity of the serotonin within the cell, and create the particular polygenic scores. And here is only 3, but now there is a very well-known approach, and I think very promising, that we can take hundreds of genes and interact with particular polygenic scores related to particular biological mechanisms you are interested in, and then it creates you ability to check particular mechanism and to have smaller number of individuals to get the effect. And the same we did for the dopaminergic activity, and that was our task. Who didn't know, so we present different stimulus, the stars, for different time intervals, and we ask the people to compare them. And normally when you compare the pairs of duration, in even if we have also them being similar, and when they're similar, certainly, and we don't allow to say they're similar, we ask, say, either the first one was longer or shorter. And generally, if you just think that it's similar, then and half of your trials, because we repeat each pair for 8 times, so about half of the times you will say it's longer, and about half of the times you said it's shorter. That's mean that you don't differentiate them. And this point is called point of subjective equivalence, so that the people don't differentiate. And if we increase the separation, you will get the more often the correct answer, so-called, like this is longer or this is shorter. But in the normal population, there is a huge distribution, and usually the first duration seems to shorten and shrink through the time. And the kind of degree of the shrinkage is can be represented as this psychometric function. So you have a kind of a shift of this point of subjective equivalence to the left. So the first duration have to be much longer than the second one, that the people will think that they're similar. So that's kind of interesting finding. And what we found, that's the model, like about neuronal accumulator, which can be related to this loss rate of this neuronal accumulator, why we have the shrinkage. And what we found, that our genetic scores relates to the activity of serotonergic system, to this subjective shrinkage. So if you have more active serotonergic system, you will get more shrinkage in this duration neuronal accumulator stuff. So, okay, I don't have it's highlighted here, unfortunately. I had it in different slides, but here you can see the number of subjects which with a particular shift at the point of subjective equivalence. So that's this subject that they have a normal or like kind of real time duration perception, and the people who is in this part of the graph have a more shrinkage of time. And by the different colors, you have the representative of the genetic groups, which has, in this case, which is a black, which has the higher activity, higher predisposition of the activity within the serotonergic gene, and in the white, you will have have a smaller predisposition to activation of serotonergic system. And you can see clearly that there's more people with the genetic predisposition to high serotonergic activity is on this part of the psychological parameters. And that's how we kind of make a model. And all the model, they supports that indeed model concentration of active serotonergic receptor relates to the kind of shrinkage of our time perception. And the test reliability for our stuff is also was also very high. And that's kind of the model which corresponds again to psychopharmacological findings. So in a particular thing. So that was the first part of the talk, which more relates to the general concepts. And now I will talk more about genetic disorders, because the genetic disorders is what we can have insights about how the human normal neurotypical people behave, how the things appears, because it's create some kind of experimental model that we can check what's going on when particular genes are mutated and oscillated, and so it's very important to study this to understand the normal behavior and novel functions. Okay, that's also a little bit of disclosure. I want to also have a question to you. What is your opinion? Does genetic means inherited or comes from parents, or is not necessarily the case? Or not? What is the example of different of not inherited but genetic? Olga, you spoils everything. I know that you know. Okay, it's hard. The audience is so small here, and the Zoom, they're probably sleeping all the way. But yes, when we talk genetics, it doesn't mean that it's inherited and it comes from parents. Although most of our genes comes from parents, but we have a lot of stuff going on and within our particular organism, and we have a lot of spontaneous so-called de novo mutations, which makes us different, and that makes allows the progress of the evolution and so forth. So, and in many diseases, including like autism spectrum disorders, this is, and the most difficult and severe disorder, they all comes from de novo mutation, because otherwise it's if it's not parents could not survive and have children even in particular with particular mutations, but still they occurs because for the evolution process, we need to create some small changes. And as I said, we have milliards of nucleotides, but and particular change in the nucleotide can be functional, and this function can be either to the bad or to the good, and some like kind of outliers occurs, and that makes our possibility to adaptation increase. So most monogenetic diseases, which is caused by particular mutation within one gene, is also called because of the spontaneous de novo mutation, because they're severe and that's more dramatic effects. There are some oligogenic effects that caused by a few combinations, but most traits psychological is polygenic that have caused by many genes. Okay, that's fine. I don't know. Okay, I can tell you about some of the most studied and diseases which we know now. So one of this is Huntington disease. It's what is important about is and interesting that it's one of the diseases which indeed has a very huge genetic components, and it's autosomal dominant. At the same time, that's inherited from parents, and it's very drastic. It's if we have the particular part of the DNA region replicated many times, the length of this replication kind of had a very high correlation of the time of your within your lifespans when you die. Like, you know, so it's seems sometimes funny, but it's not, I mean, it's indeed very strong correlation. So we, if you have many replications, you the changes in your system, the muscles and the cognitive function will decline much quicker, and there is now even kind of a formula that depending on the length, you can say, okay, you will die like 30, you will die at 50, you will die like 25 and all 70, you know. And that's very unique because we don't usually have that one-to-one correspondence. So what's happened, why we have that huge effect, which have a drastic effect on your cognitive function, and it's inherited. So it's still in parents, and it's still in our, while it's not dissipated through the evolution. Like, what do you have, any ideas why it's still? Because most severe diseases, they are not stayed long and don't stay through evolution and doesn't translate from parents, certainly if it's dominant effect. How that could be possible? Yes, indeed. That's the main thing, that usually all the drastic and severe effect they occurs already in childhood, and then it's drastic and that's prevent for living and reproduction. But here, indeed, you can live for reproduction, and even there is the particular island where the people who kind of have this disease, they live like they know like that, okay, they will die, they will have a cognitive decline quite early in life, and the early participant of the member of this island and population, they will have to take care, but like life goes on, and this particular population was very important to study, because studying this population, the researchers found the particular region of the it's the chromosome which relates to this disease, and then they even created find the particular gene and find this all the things I already told you about, this number of replication and so forth. But that's a kind of not very kind of optimistic story. So still we can't really treat it, although we know particular mechanism. Researchers still didn't find a particular cure, although I think there is a lot of studies goes on, but And now there are ethical issues about it. Do they need, like the doctor, if they find that you have this mutation and you know when you kind of die, do you need to tell it to you? Do you and what you will do then just with this knowledge if we still can't do anything with this? And but I think the situation, as you can see later from my talk, and as you can read through articles, within the last 10 years, it's changed so quickly and drastically, and we create new approaches for gene therapy and so forth. And I think soon we can identify some cue to help these people, but still it's one of the examples that we indeed succeed to identify the things, the cause and everything, but we can't still treat. And that's indeed very, very strong gene to phenotype relationship. But now I can tell you about more optimistic, I'm sorry for this changes and overlapping slides. It's phenylketonuria, I think that most of you know, but that's a much more optimistic story. That it is a disease which caused by mutation in genes, which lead to low level of enzyme phenylalanine desoxydase expressed, and phenotypically it has a decline of cognitive function, hypertony, and other problems. But it's this phenylalanine, it's metabolic agent, and although at the beginning these children, these individuals have indeed drastic cognitive decline, but when we understand the particular gene which causes this, and then we understand this function, that was easy because of this function is kind of very, very kind of simple from one side in metabolic, just using the diet, just which excludes a particular food. These people could really fully live normal life after this. And if you started early, like in childhood, it's you don't even notice that it's something different. But even in the older people, putting on this specific diet, which dissipates this particular products, helps them to improve, not to the full extent, but still. So that is, and now you don't have the phenotype of phenylketonuria in the modern world at all, but although we have these mutations, and it appears, it's one of the diseases you screened in when the child is born. That is why it is so important to screen for most of the more drastic diseases for which we can prevent the development of the phenotype. Okay, I could also tell you about complexity of genes. So you think that if you get the gene from father or mother, it doesn't matter from one side. It's was long belief about this, but appeared it's not the case. And there is a different expression levels. And I don't know, we still don't know particular signs on the parental and maternal chromosome, how it's differentiates, but as you can see from the example of these two neurodevelopmental syndromes like Angelman disease and Prader-Willi syndrome, it's caused by the same difference in the same region of the chromosome. But in one side, it's with the problems comes from the paternal chromosome, and in the one side in from the maternal imprinting. So it's depending on from which you kind of inherited the mutated gene, you will develop different types of the syndromes. And now I come to the polygenic disease, which is autism spectrum disorders. You all know, I think that the major symptoms is social communication problem and stereotypic and repetitive behavior and restricted interest and sensory issues. Again, about the history, the phenotype was described in 1943 firstly. Then the particular gene starts to occur and different animal models develops. And at the beginning, we was very enthusiastic that we find like a few genes related to autism and then can solve this problem completely. And the enthusiasm at the beginning comes from the discovering that the autism has a huge genetic component. How we can understand it? It came from the twin studies when we compare monozygotic and dizygotic twins. We can see that about 90% of the monozygotic twins will be concordant for the autism. So if one have autism, the other will also have autism, and siblings will have 25% rate of the concordance. And in a common population, you will have only like about 1% of the autism. So it's different. You can see that this is inherited and it has a huge genetic components, huger than many others in many other disorders we know. And that's why we was at the beginning searching for particular genes. But the more people look, more consortium studies, there's a huge number of patients were created. The understanding came that it will not be a few genes. It will be hundreds or even probably thousands genes which interacts and creates the complexity of the interaction which can cause to the disease. But few genes will be also kind of was identified, and through the idiopathic autism, we now can segregate the syndromic autism, which was previously within the autism spectrum disorders, but now have a particular known genetic components. And I will tell about this later. And so this is the framework how we can look at the autism. So and how we can help to understand it through neuroimaging methods and developing drugs testing on a particular organoid developed through the human cells. I think I talk about this later, but this just bear with me on this stuff. And that's what I told you, that there is a polygenetic etiology, and but one of the particular common mechanisms that might be one of the theory of the autism is the imbalance in excitation inhibition balance. And there are many genes which link to this. And one of particular gene is, I will talk about this. And here I want to emphasize that even for autism, we have animal models, but it's hard to find the particular behavioral patterns. That's why we need probably neurophysiological indexes, which can be translatable among humans and animals. So, and this is EEG. A good signal-to-noise ratio can be good translatable and can detect abnormalities even if we can't see it on the behavioral level. I mean, usually we have clear behavioral abnormalities, which is, but the changes in these abnormalities, it's harder to detect sometimes on behavioral level than on neurophysiological. And I will give you an example about auditory steady state response, which is similar among animals and humans. And if you present the particular clicks of duration with particular rate, our brain starts to oscillate at this particular frequencies. Why this frequencies is important? So it was shown that 40 Hertz oscillation and this entrained oscillation is related to the NMDA receptors, which is a key excitatory receptor in our brain. And as we talk about excitation, inhibition, balance, certainly an NMDA receptor is one of the key players in this game. What about this response in autism? That is, I think, typical kind of results, which is not only related to autism, that some studies showed great difference, like attenuated of the steady state gamma response, and in even a very small sample, as we know for nowadays, like 10 participants is a small sample. But 10 years afterwards, in the bigger samples, no atypicalities was found. Although in the middle, there was even the reduced SSR response found in the relatives of the autistic kiddos. And that's it's good, because that means that it is not related to the state of the disease, that is they like, you know, do something, but it's kind of maybe in the core of the predisposition of the causal relationship, some of the related to the why the disease is start to manifestate. So, but this result, when some is shown, some doesn't replicate, it tells us, I mean, at least me, about the heterogeneity of the sample. And that's, I will kind of show you a great example from our study that we can indeed confirm this. So again, go back to these 40 Hertz oscillations and what we know how they generate, and 40 Hertz oscillations known to be related to NMDA receptors interaction on interneurons of the alpha-bulimine interneurons with this inhibitory neurons. And the more this kind of system works properly, the gamma oscillation occurs. And there was also a mutant mice created with the switch of this NMDA receptor on this particular inhibitory cells. And you can see that the mutant cells doesn't have the proper oscillation on this 40 Hertz in response to auditory stimuli. So that's kind of a causal relationship, which we can kind of confirm. And NMDA receptors for proper function in each shunt tree is a key scaffolding elements and the postsynaptic density. And this shunt tree gene was implicated to autism for a long run. And it's one of the most cited and known gene replicated in autism. But mostly it's related to particular syndromic form of the autism. It's Phelan-McDermid syndrome. And in the population, it's about 2,000 cases described. So it's very rare. Moreover, there is a duplication in this particular region with the SHANK3 gene, and that's only few, even 30 patients described so far, and one of this was described by our group, and we have published this result, and I will show you briefly. And this phenotypes partly overlap because it has the hypotonia, mild or strong intellectual impairments, autistic traits, and particular language problems. And as I told you, that this gene is implicated in the idiopathic autism in a pretty decent percent of cases, at about 2% if we have, and about with the cases with severe intellectual disabilities. And in 15 cases, the genetic expression of this gene, of the molecular change within this particular pathway is affected. And again, the confirmation that is when you have SHANK gene mutation, you will have the difference in NMDA receptor functioning. You can see here an animal model. And here our work, which you might heard about already in the institute, but I remind you. So we had the particular patient, the girl, 15 years old, that doesn't have a strong phenotype. It's just a mild impairment, but she attends a common school. But her particular interesting stuff about her language, and that he has part of dyslexia, and he can't pronounce long words, and her speech was, if you talk about the pragmatics, was hard sometimes in creating the long sentences. But otherwise, it can be noticed it's only by the kind of experience size. Generally, she showed just probably much more infantile behavior than her typical pairs, but doesn't have any profound difference. And we, in the intellectual kind of function, he has a little bit lower, no profound MRI or clinical EEG difference. But what we have found that this RSSR response on 30 Hz were completely absent at her. So she couldn't oscillate, her brain couldn't oscillate in response to auditory stimuli on the 30 Hz, although she heard the stimulus somehow, but she couldn't differentiate this frequency at all. And that might be kind of how this decrease in her auditory cortex resolution might cause her the problem and the delay in the language acquisition, which she showed through her childhood, and the problems also to deal with some longer and new words acquisition. Now later, then we also work with the Phelan-McDermott syndrome, those which has a little bit higher representatives within the population. We found 7 cases with this, and we also confirmed that even they have both duplication or deletion of this within this SHANK3 gene, this 40 Hz response is fully absent. And these people also have some problems with language acquisition, and even more profound. So how it's related to idiopathic autism? So as a final kind of chord in our story. So we have studied the idiopathic autistic children, have a group of more than 60 participants, but our geneticist found that and 8 of this, they have a particular small duplication within the SHANK3 region, small micro duplication within this CNV's region, as we already talked about at the beginning. And that was the MG study, but it doesn't matter. This 40 Hz oscillation can be recorded both on MG on EEG. And that is how we can see this 40 Hz oscillation on time frequency analysis. But it's, I mean, just completely similar what you kind of, if you'd have the Hilbert transformation, which I was showing you previously about this. What is interesting here, we just for imaging, we can a little bit better to identify from which the signal comes from. And as you can see, it's auditory cortices. And in red, you can see this group of idiopathic autistic kids which has a particular mutation within the SHANK3 gene, and you can see they have it absent. But if you look at the green line, which represents the other autistic kids without this mutation, they didn't much different from the typical developing group. So our auditory steady state response can help to segregate the groups and within the idiopathic autism who didn't know about any of the particular mutation and the fields affected. And while we could differentiate it, we can already tune the particular treatment for them, which provides a very great opportunity for personalized treatment of these kids. So that is basically what we can have on the fundamental level of the science. So we found that this particular gene relates to our SSR decrease in the particular frequency, high frequency, which is related to binding between different regions, and also binding of different features on auditory signal, and was related in previous study to the detection of speech in noise and detection of small gaps between the auditory stimulus, which is important to process the speech information. And at the molecular level, we think that we have a strong evidence from the previous literature that this relates to hyperfunction an MDR receptor on being positive interneurons. And at the behavioral level, it's caused, as I said, particular speech problems, which relates also to autism, but is particular subdomain of it. It's not purely relates to autism at all. Okay, and so probably just to conclude about how we can move. So that's one of the particular example how we can segregate the autistic kids or other population. For example, we work now on the time perception and serotonergic gene, and we can already try to segregate this depressive people of the particular subgroups. But what is going on generally? So we have to think of the forward and reverse genetics. So forward genetics is, as an example, we have with Drosophila, for example, at the beginning when we kind of induce mutation on this population, but how they induce it, just particularly the teratogenic factors. So they just look at the population and segregate the particular phenotype. When you segregate the particular phenotype, then you can look at the genes. So they don't, at the beginning in the forward genetics, you kind of increase the possibility for the mutation, but look at the phenotypes. Reverse genetics is much more kind of targeted approach. So if you already know gene, you can now induce this mutation or induce the difference, induce the change in the genome, and then you can see, okay, if you do particular change with the genome, what is the phenotype then? And that is much less work from one side. I mean, it's technically more advanced, but it's more straightforward. That it's not like you look at the many phenotypes and try to segregate them and then particularly related to the genes. And here is my question to you. So what do you think, where the first genetic therapy, genetic alternation in humans occurs like officially, or it haven't occurs at all yet? It's our future, or when was it, if it was already done? About 20 years ago. Okay. In what do you know particular, or if there's any other opinions? Do we already do genetic alternation in humans officially? Yes. Spinal muscular atrophy, I think you was thinking. Yeah. Yes, indeed. It's, well, thank you very much. Yes, indeed, it's already present, and it's yeah. And the first article about this, it was a little bit even earlier than you said. It was 1990 when the first gene transduction occurs. But most first application of this was for metabolic disease, and certainly the disease which causes death to the patient, which is because it's experimental. yeah, you know, you do drastic change in your genotype. You don't know how to reverse it at all. You don't know what's going on. So to try it in human, it's a big deal. So, and you have to have a lot of ethical concerns, but in only the case when you know, kind of, otherwise the people die anyway. And there was several good examples, and most of them the good at the beginning, but there was also a lot of setbacks occurs later on that's mostly related to oncology. But I mean, it's going on already for tens of years, and in the different diseases, and we apply it, and usually how we do it now, and mostly the idea was the same as in previous. So we do create the particular adenovirus or other virus which goes through to your DNA, and using the particular markers, it's find the particular region in the DNA and change it by the particular mechanism. And I think in our institute, there are people who knows a lot about it. For example, Pavlova, and she's doing, I think, very, very interesting research here. But the idea that it's present, we have it, and we can apply it. And now, and so there are several diseases, even the syndromic diseases related to autism now. It was like before pandemic, we should have one first clinical trial for Rett syndrome, and now for Angelman syndrome is also planned, which will particular change the DNA in this. But due to some other problems with the other, for example, spinal muscular atrophy disease, with already FDA approved and do this genetic therapy, we do have we do have ideas. It was delayed, so I don't know exactly when it starts, but all the tools mostly are approved and prepared. One of the things which is probably even more promising and more safer than the change in particular DNA structure is change in RNA in an expression level. And because, for example, as I tell you, if you change the DNA, it's probably again a couple of years or probably 10 years should have passed that we understand how to reverse it. But for RNA, it's easier. You try, you see what's going on. If you like the outcome, go on. You can continue to use this drug. But if it doesn't work good, so you can stop, and then the DNA stays the same. And so we change just the expression level. Yeah, but that's what is, I think, even more promising, because most of our studies now and all the drugs we should try and genetic therapy we tried, the first, certainly we need to do clinical trials on animals. But now the new era, I think, comes, and the souls will fully develop, and we can create patient-specific organoids based on induced pluripotent stem cells, and you can take the stem cell from the not even stem cells, you can take the somatic cells from the individuals, then you kind of reprogram it to the stem cells, and then differentiate to particular tissue you want. And certainly more interesting people is in neurons, and even we can create the organoids and test new drugs which particular suits to particular personalized stuff to particular people. So I think for this I have to stop and welcome your question.